for introduction, the process through which epithelial cells convert into mesenchymal cells is known as epithelial to mesenchymal transition (EMT). Then, epithelial cells produce the epithelial tissue that covers an organism's internal and exterior body surfaces. These are called polarized and create extensive cell-cell adhesions with one another, including adherent junction and tight junction.
An epithelial mesenchymal transition (EMT) is a biological process that allows a polarized epithelial cell which normally interact with basement membrane via basal surface to undergo multiple biochemical changes. The completion of EMT is signaled by degradation of underlying basement membrane and the formation of mesenchymal cells that can migrate away from the epithelial cell. The figure shows the passage of cells through EMT. The epithelial and mesenchymal cell marker commonly used by EMT. Co-localization of these two cells of distinct marker define an intermediate phenotype of EMT, indicate cells that have passed only partially through an EMT. Detection of cell expression both set of markers makes it impossible to identify all mesenchymal cells that originated from the epithelium via EMT, as many mesenchymal cells likely shed all epithelial marker once the transition is complete.
Next is the loss of epithelial marker and function. And during EMT, epithelial cells lose their cell membrane epithelial marker and acquire mesenchymal marker and metastatic traits. The EMT is a biological process in which a non-motile epithelial cell changes to a mesenchymal phenotype with invasive capacities. And this phenomenon has been documented in multiple biological processes, including embryogenesis, fibrosis, tumor progression, and metastasis. The hallmark of EMT is the loss of epithelial surface markers and most notably E-cadherin. Next is cell-cell interaction, when during the initial stage of EMT, epithelial cells decrease the expression of essential protein involved in homeostasis, and particularly those involved in cell-cell interaction and ECM cell exchanges. Furthermore, the cell adhesion loss has been linked to migration, invasion, and eventually dissemination inside the body. The four major adhesive and communicative junction, AJS, tight junction, TJS, gap junction, GJS, and the ensom, and all diminish in such situation.
For the gain, mesenchymal marker include the transcription factor production and cell function. For transcription factor production, a large number of factors can be described for their roles in embryogenesis and have been associated with EMT. SNEL1 and 2, TIS1 and 2, and ZIP1 and 2 will be deciding the factor for roles of the cell type. SNEL gene is usually a strong repressor that is given a negative regulation of the EMT marker, claudins, occludin, and E-cadherin. This will regulate by FGF, hepatocyte growth factor, ringless integration site. Different types of cancer will be increased the SNEL expression and down-regulate other epithelial markers such as demospakin. These 1 and 2 are basic helix loop helix transcription factors that were found in many cancer and correlated metastases that are overexpressed. These two proteins can be controlled by the signal factors of FGF and EGF. Mutation that occur in development disease is usually expressed in mesenchymal and neuronal progeneration cells that can be found in Z1 and 2. It's can regulate by hypoxia-inducible factor. Stem cell marker like KLF4 can be increased and also repress the MIR200 family by activate the MMP family members. For the cell junction, due to the lack of the intracellular connection and specific enzyme regulation may result in the ECM repression. Organization and localization of fibroblasts show a similarities with the mesenchymal cell, and it can be seen example of cytokeratin had a low level in mesenchymal cell compared to the epithelial cell, while E-cadherin were specialized in express and EMT epithelial marker is well categorized, and N-cadherin is used in measure of mesenchymal marker. Cells that had ability to migrate can inhibit the E-cadherin expression as it coincides by increasing N-cadherin expression, as shown in case of benzophenols 1 and 4 OT-phenols, and in human ovarian it was first described the role of this marker when wound healing and promotions of fibrosis by aiding the differentiates between resident tissue fibroblasts into microblasts.
Okay, for my part, this role of EMT as potential marker in food toxicology. Okay, for the teratogenic effect, epithelial to mesenchymal transition (EMT) happens during plasminogenesis, early fibrosis, and late metastasis stages. Next, EMT, on the other hand, is a well-known multi-step physiological process that leads to embryonic growth and tissue healing. Lastly, it's now possible to determine the teratogenic and pro-tumoral effects of food follicles and ages at the same time, thanks to the use of EMT biomarkers as a predictive tool. Thank you. Next, for the pro-tumoral effect of food contamination. The epithelial to mesenchymal transition is a complicated biological process that has long been linked to cellular phenomena like transformation, division, and transdifferentiation. EMT is an either consensual, trustworthy, and economical approach for assessing the effects of environmental pollutants. Besides, the consequences of foodborne exposure to chemical pollutants or the high amounts in the human diet, such as BPA and pesticides, may be particularly fascinating when using EMT as a toxicological technique. Thank you.
Type 1 EMT is associated with implementation embryonic granulation and give rise to mesoderm and endoderm and to mobile neural crest cell. The primitive epithelium, specifically the epiblast, give rise to primary mesenchyma via an EMT. Unlike the type 1 EMT, the type 2 EMT is expressed over extended period of time and can eventually disrupt an affected organ if the primary inflammatory insult is not removed or attenuated. Type 3, the secondary epithelium associated with many organs, can transform into cancer cells that later undergo the EMT.
Okay, for the Q&A session, why is epithelial mesenchymal transition important? This is because many tissues and organs in the developing embryo, as well as various embryonic events such as gastrulation, neural crest creation, harmful construction, and etc., are dependent on EMT and its reverse process, which is MET, mesenchymal epithelial transition. That's all for me.